And our final group are nucleic acids. Okay, nucleic acids, also known as nucleotides. So we're specifically going to talk about DNA structure. That's what nucleotides are used to do. So think nucleus, that's where the DNA is, nucleotides. So each nucleotide has three parts. There is a sugar connected to a base and a phosphate group. Okay, so cyclic pentose sugar, notice these are my pentagons. So it's 5 carbon sugars connected to a phosphate group and some type of aromatic base. Okay, so this is an example of an entire nucleotide. This is my 5 carbon sugar. And on carbon 1, we connect our base. This is one of those aromatic bases. And on carbon 5, we connect our phosphate group. And that is a single nucleotide. Okay. So structure-wise, right, the pentose attaches to the base of carbon 1, phosphate attaches to the base of carbon 5, and then those phosphates link together to make our DNA backbone. Phosphate sugar bonded to phosphate sugar, bonded to phosphate sugar, and you have those bases hanging off. Okay, so what's the difference between DNA and RNA? It is one little oxygen. Okay, DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So all we have the difference between these two saccharides or monosaccharides is one little oxygen. Okay, now that one little oxygen is really important because deoxyribonucleic acid can't go outside of the nucleus, whereas ribonucleic acids can go outside of the nucleus. Okay, so those are the difference between our two 5-carbon structures. So the bases that are attached to DNA and RNA are adenine, guanine, cytosine, thymine, and uracil. Okay, in DNA, there's only A, G, C, and T. In RNA, there's only A, G, C, and U. So thymine and uracil are kind of interchangeable in sequences, but DNA only has thymine and RNA only has uracil. So these are the bases that can hang off of your chain. So when I'm talking about bases, it's this thing up here. Okay, so base pairings. When you think of DNA, you think of a double helix, right? One strand and another strand, they match up, and then you get your double helix. Well, what is that matching up? They're actually hydrogen bonds between the two bases. So G and C attract each other, and A and T attract each other. G and C actually have 3 hydrogen bonds, whereas A and T only make 2 hydrogen bonds. And you'll notice each of these pairings is between a 2 aromatic base and a 1 ring aromatic base. So G and C, this is G, it's a 2 ring aromatic base attracted to a C, which is a 1 ring aromatic base. Same thing with A and T, but it's only 2 hydrogen bonds. This is a 2 ring aromatic base attracted to a 1 ring aromatic base. Okay, so those are our base pairings, and you can tell what the DNA really says based on this code. Okay. So how are those all bonded together? They are bonded through the phosphates and the sugars, right? So you have a phosphate part, and on this one, it's going to attach to the sugar. Phosphate from this one bonds to this sugar, and so on and so forth. And the bases hang off the side. So this is an example of a strand of DNA. On one end, you're going to have an extra phosphate group. That's called the 5' end. And on the other end, you're going to have an extra sugar with no phosphate. That's called the 3' end. That's where the ribose is. So linking these nucleotides, similar to a lot of the other bonds we've been making, are you remove a water and you get a bond between your sugar in this case and your phosphate in this case. Okay, so you're taking a water out to get those to bond. And so then we've got our DNA strand. Now, what do we do with our DNA strand? Well, our DNA strand has what's called codons, and every 3 bases codes for 1 amino acid when we're building protein. So the entire purpose of DNA is to give us the sequence to build a protein. So this is a codon, an AUG, for example, codes for 1 amino acid that goes into a protein. So a codon is a 3 nucleotide sequence. 1 codon codes for 1 amino acid to build a protein. For example, if I have A and then G and then T, that tells me a serine goes into the protein. This is universal for all living things. So this is how we figure out DNA things that match, right? We're like whatever percent similar to apes, and that's because we use this same coding system. That's how we can figure those things out. So I talked about the DNA double helix before, and I'm going to talk about these base pairings a little bit more. That's how we get the helical structure. These are our hydrogen bonds between a G and a C, or an A and a T, and that's what gives you your double helix. Okay, so what we need to do is we actually need to take apart these DNA molecules in order to figure out how to make the proteins. So taking apart DNA molecules to figure out our proteins is similar to DNA replication. So first of all, I'm going to talk about DNA replication, and then I'll get into protein synthesis. So DNA replication is kind of its own thing, and that has to occur whenever we make new cells, right? I have to actually make new DNA strands to be in the nucleus to be able to produce things that we need to survive. So we have to separate the strands, then we make complementary strands, right? So that I have this code and this code, I make complementary strands, and now I have two identical copies, and then those can be used in new cells. So that's DNA replication. Whereas when I talk about protein synthesis, which is really what I wanted to talk about today, I do what's called transcription and translation, because, right, the DNA codes for creating proteins. So the DNA, which is in the nucleus and can't leave the nucleus, is first transcribed. So you can think of like you're transcribing into a different language, the DNA into RNA. So DNA gets transcribed into a complementary RNA strand. Then RNA can leave the nucleus. So it's transcribed or put into a different language and then transported into the cytoplasm. And in the cytoplasm, mRNA, it's messenger RNA, a specific type of RNA, then links up with a ribosome, the thing that creates the protein, and we put together a peptide chain. So it reads the mRNA sequence, mRNA is messenger RNA, and it tells you what amino acids need to be bonded together to create this protein. And so that's transcription and translation. And I'll really get into biology here, but the ribosome is the thing that reads those codons and puts together the amino acid sequence. So this is what it looks like. Okay. Got my mRNA strand that came out of the nucleus. It's already been transcribed. Now we are going to translate it into protein structure, which is completely different. So transcribing is just putting from DNA to RNA. Did I say that right? Yes. And then translating is creating a new thing from that mRNA strand. So a ribosome has two subunits come together. They stick on your mRNA and they read the mRNA, those codons, and it tells it what amino acids to bond together. Okay, so I have a growing protein over here. It's reading the mRNA, and then when it's done, I have a nice new complete protein. The ribosome can detach itself from my mRNA molecule, and then it can go read another mRNA molecule and build another protein, and so on and so forth. So the DNA turns into mRNA, and then we read it to get our nice new proteins that can form their secondary and tertiary and quaternary structures that do things in the body.